Okay, so now that Simon has found his place, we can start. Now, welcome to the second session. That's the short topic session. I'm Kaspar Rufibach. I moderate this session together with... So she's Elena from Female. She'll figure it out with the microphone.
Yes, now it works. Yes.
Yeah, just to reiterate, everybody has to sit, otherwise they'll kick us out of the building, and they're serious about it. So then this session will be very short. Anyway, so we start with the first talk, and the first question. That's Judith, who worked together on this with Annabelle and Alex from Roche. And please, Judith. You can move the slides with this one.
So how do I move it? Good morning, everyone. As they said, my name is Judith. I am happy here to present on behalf of my colleagues Annabelle and Alex our questions for the regulators. So we are working in a clinical trial which is currently recruiting, is ongoing. And this trial, we are recruiting patients in a disease which is in early stages of neurological disorder. And apologies, I cannot give more details. So we are recruiting approximately 500 patients in the early stages of this disease, and we expect a mortality rate of 1%. And this is according to literature. So we know that very few patients will die in our trial. For simplicity, for our estimate, we're assuming for this presentation that we have only an intercurrent event. The intercurrent event is death. And we have two treatment arms, the treatment arm A and treatment B. We have two endpoints. The primary endpoint is a time to event in which we are measuring time to a progression of certain events. And we have a key secondary continuous endpoint in which we use the same scale as the time to event, and this scale is assessing the symptoms of the patients and how much they progress into the disease. So our proposed strategies for handling the intercurrent event of death are as follows. For the time to event, we selected a composite strategy in which we assume that death is counted as a progression event. And for the continuous endpoint, we chose a hypothetical strategy in which we are allowing the statistical model to answer the question or to estimate the treatment effect of what would have happened to the patient if the patient had not died. It's a strange question, but so we submitted our statistical analysis plan to the FDA, and the FDA gave us this feedback. For the time to event, they said that the composite strategy would not be appropriate for handling deaths unrelated to disease progression or a study. Well, I will leave it in disease progression, sorry. And so when we saw this feedback, we replied to them that we would like to keep our selected composite strategy as the main one, because we consider death as a poor event, and this will be reflected in the composite strategy. And in trying to be collaborative with the FDA, we added a supplementary analysis of a hypothetical strategy in which we will censor deaths at the time of the event. Then the FDA gave us the feedback that for a continuous endpoint, a hypothetical strategy is inappropriate for addressing the intercurrent event of death because it is clinically implausible and does not reflect real-life situations. And I agree, but this is where my questions go to the regulators. So the first question for the regulators in the time to event context is, what is the importance of differentiating unrelated disease progression deaths, knowing that we have less than 1% in our trial, and when the composite strategy is conservative and penalizes all deaths as events? That's the first question. And for the second question is in relation to our continuous endpoint, in which for any estimate strategy will be implausible for estimating the treatment effect of a person who died. So which estimate strategy do you suggest for handling deaths in this context? Always keeping in mind that it's only 1% of deaths.
Yeah, so how we run this session now is that all the regulators who want can comment Judith can ask follow-up questions. The audience can also participate. It's probably difficult to take questions from the Q&A, but you can also post there if you are joining. No, if you're joining, also most likely we just participate with the audience. Also, all regulators in the audience can participate. But maybe we quickly just introduce the panel so that everybody says their name and which agency they come from, because then it's nicer to have an interaction when you know with whom you are speaking. So Florian can start, and the mic is probably not on, or is it? Yeah.
Thank you. Yeah, my name is Florian Kliemüller. I'm from the Austrian agency.
Frank Pestavi, EMA and MEB.
Andreas Brandt, BfArM, one of the German regulatory agencies.
Benjamin Hofner, Paul-Ehrlich Institute, the other German agency.
Eva Skovlund, Norwegian Medicines Agency, or Medical Products Agency, actually, and CHMP.
Lukas Agere, also the second German agency, Paul-Ehrlich Institute.
Kit Roes, MEB, which is the Dutch agency, like Frank, and chair of the Methodology Working Party of the EMA.
Khadija Rantel, MHRA, UK agency.
Bergrún Magnúsdóttir, Icelandic Medicines Agency.
Okay, thank you. And then let's go to the discussion. So who wants to start? Andreas, please.
Yeah, I think first of all, it's what we always regret is that we are here a panel of exclusively of statisticians who discuss what is the appropriate estimate, because I think we should have mainly also clinical involvement here. But actually, to the questions, I think for the time to event endpoint, I do not really understand why it would not be appropriate to use death as an event, because I mean we also do it for other diseases. Yeah, of course in oncology for PFS and also for cardiovascular diseases, where we also have lots of discussions and trouble with CV events or non-CV events and how this is educated. And especially here in this context where we have only very few deaths, I think it would be here the best solution to use this as an event. I mean, of course, for the continuous endpoint, it's more tricky. But here, it depends on the endpoint, but in some sense, you could even argue that, you could assign a value to the patients who die. For example, if it's a functional endpoint, you could even argue that death means a loss of all function and use some kind of, so to say, interpret it as a treatment policy. But of course, or you could also use a composite. I mean, for analysis, this maybe even be similar to considering 0 or whatever is a true value, because then maybe death are outliers and it may be more natural to use a rank-based analysis. But although this, of course, causes problems with interpretation of the treatment effect. But
yeah, sorry, let me just, I forgot to mention for the continuous, we proposed a supplementary analysis in which we are assuming a composite and we are selecting a priori a cutoff on that represents the worst event in the scale. But what I find difficult with that is that it's a longitudinal trial. So if the patient that die halfway through the study, then you fix that cutoff and carrying on use it through the remaining of the trial, which is very peculiar.
Eva, please.
Yeah, I must admit I found this a bit strange. I was thinking with the time to event endpoint, I would be totally happy with the composite endpoint. So that's my simple one.
Florian?
Yeah, really. No, I think with the composite endpoint, I think one thing that one needs to be a bit careful with and needs to make clear is that in the end, a result on the composite endpoint is not interpreted as an effect on survival. So because now you have also death in there as an event, and there should not be any misunderstanding that if this is just that there is no effect on survival in there.
Okay.
And that
I
can be
get-
made clear by
Yes.
just showing that these rates are not different. And if you want to have a claim on that, then
Okay.
you need you need additional evidence. But I think that is a concern that I've heard, that if you put like mortality into the composite endpoint in there, one may interpret like evidence for a treatment effect that the drug also works on survival.
Yeah. So let me just point out that on the death, I mean, the question is differentiated unrelated disease progression deaths. So there is here, the FDA made a separation between the disease-related deaths and the unrelated ones. So you are referring just now to the unrelated deaths.
In the end, you come up with a composite endpoint that is like time to some sort of progression or death. And if you say we have an effect on that, people may misinterpret this, that you have an effect on, on mortality.
Yeah, but thank you for your other comment.
Lucas, please.
Yeah, so the part of the question was on differentiating unrelated deaths or not. And while I fully agree with what Andreas said, that we are only the statisticians and can only give part of the perspective on the estimate, and the other perspective would as well be very relevant. I think we should acknowledge that even though the expected mortality may be small, there would also be uncertainty in the differentiation, whether you get the differentiation right. Is it related or unrelated to the disease or to the drug? So then to be on the safe side, I would also prefer to include all deaths here. But yeah, these are my considerations on this.
Oh, thank you. Sorry, can I?
Yes.
No, thank you. I think that's a good comment. The point of differentiated and not differentiated, in the study, we didn't set up the study to differentiate. So the only way that we could think of doing it is establishing an adjudication committee where experts outside the trial will determine if the death was related or not related. But that adds complexity into the trial.
Indeed. So this is one of the experience that I made over the past years, that this differentiation can be helpful, but there will always be some sort of uncertainty with it.
Thank you.
Rather a question than a comment, but maybe it includes a comment. Did you think about competing event analyses for death? I mean, if you are not interested in an effect on death, and if I got the question correctly, I mean, it's quite abstract. You mentioned it's early stage disease, so I would not assume people to die of the disease, at least on a frequent basis. I mean, 1% might even be a high estimate, but I don't know. But did you consider it to be an intercurrent event as well? And what would that mean? Or do you assume that, I mean, most of the deaths are related to the disease? That's what we do in oncology. So yes, we might have also other death cases, and we ignore them because they, in relation to the overall number of deaths, are very small. And then we use overall survival, and we ignore the source, the reason for people dying. But this depends a bit on the fraction of death adjudicated to your disease or unrelated to the disease. And maybe that's an important question, even if it's very low. And in principle, I agree to the rest of the panel. In principle, the estimate you proposed seems to be acceptable, at least from a regulatory perspective. Whether it's the relevant question for patients to be answered is a different question maybe, but there I would assume it doesn't really matter. I mean, if the effect size is so, or the number of deaths is so low, it doesn't really impact the estimate.
Thank you.
But maybe the question for the competing events, did you consider that?
No, we didn't consider that.
I mean, it's a question of whether it's sensible to consider it a competing event. I cannot judge from what you provided in the slide.
Yeah, no, but we didn't.
Hello? Yeah. I just have one comment regarding the statement that the composite strategy is conservative. I wouldn't necessarily agree to that, because it depends on, I mean, if there are more unrelated deaths in the treatment, in the placebo arm, then it would not be conservative. So it would depend. So I think this statement should maybe not be there. But if the rate is very low, it might not matter so much, but yeah.
Yeah, okay.
Frank, please.
The second question shows that there is still a tendency to put the focus on whether estimation is feasible before there is a discussion on the estimate. I think it should be more separated. And in E9 revision 1, we say that we should challenge the choice of estimate only if there are issues about the estimation, but first there should be a strong discussion on estimates. And there is a big tendency to think first about, or to think already straight away about whether estimation would be possible and how easy it's going to be before really focusing on the discussion of estimates. And that's what I read in the second question. And maybe a remark, I was a bit disappointed with this use case because it's a bit too easy to answer if the frequency of death is less than 1%. If you had said 5 or 10%, we would have had a more difficult discussion.
Yes.
This was an easy start for the short topic session, right? Did you get what you wanted?
Yes, thank you very much.
Right? Then okay, one comment, David? Yeah.
Actually, I wanted to ask this question, Frank, if 10%.
Thank you, Judith, and thank you to the panel for the answers. And as you say, it was fairly straightforward for question 1, but I'm not sure if it was so straightforward for question 2. So maybe if we could just challenge you a bit. In terms of for a continuous endpoint, there have been papers written saying what you should do about this. In terms of statistically, or you can use a clinical judgment about what you might think of an extreme value. But challenging you even further, when you think about, obviously, you know what the neurological function is in someone who's died, but is that really the question that is of interest here?
It's definitely the more difficult question. Somebody wants to give a short answer before we move on? No?
I think Andrea said it, if you die, there's loss of all functions. Then how you translate that into this low score, that can be done in, I don't know, 10 or 20 different scenarios. And that's where it's difficult to choose the one that you want to derive your decision making on. And here, maybe it's not so much about decision making, but more about the size of the effect. But it's more of a clinical question, so maybe I can hide behind the clinical thinking.
I think that there's also again the difference in that question. I think unrelated death might not be accounted in the same way as related death, but then the difficulty of adjudicating is quite prominent. But again, with 1%, it doesn't really matter, I assume.
How about any opinion on the while on treatment or while alive in that case estimate?
You saw everyone shaking their head and murmuring. I think that's usually not the estimate we are tackling, and I think but I can give it to others. I would not be very much in favor. Is that really the question we want to answer? If I die of the disease, regardless of treatment or because of the treatment, or I don't know, then yes, I want to know that. I think that should be accounted in the endpoint.
So we have 2 minutes left. Mona and Mark.
Thank you very much. Just a quick comment to Judith, actually, because I think it's difficult for me to understand why, in the first place, it was suggested to use a composite strategy for the time to event endpoint by a hypothetical one for the continuous endpoint. It almost sounds a bit like, because it's easier to handle from a statistical perspective, we sort of propose that, but what is the rationale for wanting to account for death in a different way just because it's a time to event or continuous endpoint?
No, we discussed that point, and it's a good one. We try to align it But I really struggle with what I said earlier. I mean, establishing a composite strategy in a continuous endpoint. The only way that I found to do it is in a paper which has Michael O'Kelly and other people, and they suggest that you select a priori a cutoff, which we could do that, and we proposed that for a supplementary analysis. We selected a priori this cutoff and then use that value until the time of the treatment period.
Yeah.
So I don't feel comfortable using that as a type of strategy for aligning the composite in the time to event and the continuous endpoint, and that's the reason.
I certainly understand that, but I think we are running the danger of changing the clinical question or how we want to capture death out of statistical convenience. And it goes to the point that Andreas Brandt made at the beginning. I think it's very important to first discuss, is it disease-related, unrelated? Does it tell me something about the disease or not? And then we will fix the statistical challenges, I think. But to actually have it in this way to already propose it differently, I find that a bit challenging. But then also I wonder if we would express the hypothetical differently by saying, what if nobody in the trial had died or something, instead of saying, what if this particular patient didn't die? What if we would express the hypothetical differently or express it in a principal stratum way? What if we only had randomized patients that didn't die for disease unrelated reasons, let's say? Would it be more acceptable or not? So I think it's also maybe that they react to the continuous one because it's worded in a very implausible way, but maybe we could actually phrase it a bit differently.
Yeah, okay, thank you.
Yeah, I think that was a very good comment.
Nothing to add.
We're taking one more, Caspar? Okay.
-2 minutes, Mark.
Yeah, I'll be quick. So Mark van der Meulenbroeke, UCB. I'll just share a quick experience from my previous employer without giving away too much. In this context, we did actually have a trial where we had 2 arms, missing endpoints, not because of death, but missing endpoints. And it was FDA who actually did not agree to our proposal for handling those and suggested that we would plug in the worst possible value for the missing endpoint. Now, this was a scale with a wide range, and the worst possible value on a change from baseline endpoint would be, ridiculously out of scope. And we mentioned that, of course, especially because we were expecting more such events on placebo, which goes back to what Bertrand said. It's actually anti-conservative to plug in these estimates. Still, FDA insisted that this would be the way to go, and so we did it. It's very astonishing, and of course we had additional analysis, but this can happen.
Thank you.
Thank you. I think now we move to the next one. Tobias, please.
So thank you, Judith.
So we have a second estimates question, and then the rest we save, I think, for this afternoon. So there's only 2 on estimate, but now we have okay, great.
All right, thank you. I hope you can hear me okay. So my name is Tobias Mütze, and I'm presenting on behalf of the ERWG estimation subteam. And it's on estimation, not on estimates.
Yeah. Okay.
You see the working group members here, and some are even in the room. So we are in a treatment policy setting, which means that the data we observe after the intercurrent event is relevant for the estimate and the analysis. So for example, for subject 1, they have the intercurrent event, and we observe the endpoint, and we use this in the analysis. However, for subjects 2 and 3, they have the intercurrent event, but then we do not observe the endpoint. These subjects withdraw from the study. So we have a missing data problem. And that's essentially in clinical trials unavoidable. Even with best efforts, we can maybe reduce this a bit, but it will always be there. So to estimate the treatment policy estimate properly, we have to appropriately handle the missing data. And one way that often does not do this appropriately is using a standard MRM approach. Instead,
what we should do is impute the missing data. And the two most common approaches are a reference-based imputation, such as jump to reference, copy increment reference, or copy reference, or retrieve dropout models. And in the working group, we've recently published a paper where we compared the different imputation methods. And a key finding of our work is that the methods differ in their clinical assumptions and their statistical properties. So for example, on this slide, you see a key outcome of our work, and you see the comparison of retrieve dropout methods and reference-based imputation methods. And really, the takeaway is that the clinical assumptions and statistical properties are different. For example, the retrieve dropout models, they assume that missing data after the intercurrent event is similar to observed data after the intercurrent event within the same study arm. On the other hand, reference-based imputation methods assume that missing data after the intercurrent event is similar to observed data from the reference group. So we have to make a choice of the clinical assumptions and also justify this. And in terms of statistical properties, they also differ. For example, the retrieve dropout methods, they have usually a very small bias in realistic scenarios, and they make no assumptions about the treatment effect. However, to really fit these models and use them for the imputation, you have to collect a lot of post-intercurrent event data. And if this is not done, you can't fit those models, and you really inflate your standard errors. On the other hand, the reference-based imputation approaches, they control the type 1 error rate. They don't inflate the standard errors. However, they make assumptions about the treatment effect for subjects with missing post-intercurrent event data. And if we deviate from these assumptions, then we have a biased estimation. So that's just setting the scene. And now we come to our questions. So we have two questions. The first one is, when we are estimating a primary estimate that adopts a treatment policy strategy, is it acceptable to use an analysis approach that imputes the missing values using a reference-based imputation method if we can clinically justify the assumptions? And the second question is, if it's unclear which assumptions are appropriate for the missing data imputations, which principles guide the selection of the imputation model? For example, how important is type 1 error rate control, a conservative bias for the treatment effect estimate, the bias variance trade-off, and clinical plausibility? And then sub-questions are, what's the priority order of these criteria and anything else that might be important that we didn't list here?
You can see that the guidance has been 2 slides and 2 questions. So there is a lot of packed into the slide so that these guys cannot read the things there anymore. They're following the guidance, so that's good. So who wants to start? Berkram, please.
I have it on. Well, the first question is the answer simply yes, if it's clinically justified. Regarding the priority, I mean, there cannot be any general rule. It all depends on the size of the bias, the size of the standard error. So it would need to be somehow quantified to be able to answer the question. It could be no priority all over for all cases. Sometimes it would make most sense if the intercurrent events are such, it would make much sense to impute some failure. Regarding the comparison of the retrieved drop model and the reference-based multiple imputation model, well, the retrieved dropout model has less bias and would be maybe preferred, but it's very seldom that we maybe can use it because we have too little data when there is this intercurrent event. I don't know, so it would be nice, but seldom that it can't be used. Others?
Who wants to speak, please?
Also kind of grabbing the opportunity to say something, because these two cases are not entirely unrelated, the previous example and this one. I'm assuming here that the question is against the background that this is the estimate and the strategy for answering the primary question on efficacy. That was my problem with the last example. There were two analyses proposed, and I missed the precise articulation on which additional research question is answered by the longitudinal analysis. And that will actually drive, at least from my perception, what your strategy would be. And then indeed, I heard Emmanuel mention it, something like while alive might actually answer a relevant research question. So I think, but here I'm assuming
primary
it's the
endpoint,
primary,
efficacy.
it's the primary endpoint.
Yeah.
Then the first remark I always make in this setting, don't take for granted that you don't have an outcome because people drop out of the trial. So the first discussion is get it down to the 1% that we had in the previous example. And then our problems will, yeah, you laugh, but there are actually academic groups who are able to do so. So I think it's a very, very serious message, because we are solving a problem that can be partly solved by execution of the trial, by complicated statistics. So I think we need to constantly be aware that we are doing so and reduce the real problem. So that's I think one important message here. The second one, and that's where I'm not inclined to immediately say yes to one. I think anything what we do here that's sensible and does something different than the usual analysis makes assumptions about observations we don't have that necessarily cannot be checked with data, because otherwise we have methods and we are. So this will all, in my view, always be a difficult discussion. So for me, the clinical justification is kind of the starting point. So if that's still there, you're quickly done. But subsequently, what will work in practice in absence of having data to verify your assumptions against? I think there's not a clear answer. And I think so all of the criteria are necessary. And I would very much like to, let's say, take the order of priority post hoc. So that's, because I think it's not that easy to define upfront, independent of very detailed knowledge on the particular case. But I do think it's important it's highlighted in this way. So actually doing the exercise of approaching the problem and presenting it in the way that we actually have a view on what's happening here and build credibility in the assumptions we are making. I think that's a crucial part of what you're proposing.
You want to comment? No? Okay. So Benjamin or Andreas? Yeah.
Yeah, I think with regard to the first question, of course, I agree to all what Kit just said. But from a practical perspective, actually, this is what we recommend very often to do. And of course, I mean, you have to make assumptions. You have to check these assumptions, but to go and also to justify these in advance. But I think from a practical perspective, this is where this is what I would agree when I'm asking the scientific advice. If there are no specific reasons this not to do so, this would be more or less my default for the primary analysis. And actually, my concern would always be with retrieved data that we don't have enough of these. And so I'm very happy that you confirmed this with your research. And I think with the second question, I don't think it's easy to disentangle all of these issues. And I think, as Kit said, we always need assumptions that we cannot verify. So I think there it will be very important to have some sensitivity and analysis and to get the complete picture. And hopefully, if we don't have many missing data, there will be only minor deviations.
Maybe a follow-up question. Do you think it would be appropriate or possible to say we do an RDO imputation, like a retrieved dropout imputation, if we have enough data and check this before database lock in a blinded way, and then maybe decide to step down to a reference-based imputation?
Yeah, actually, this is a very good question, but I mean, maybe if it's really fully pre-specified, but I think it's difficult to say in advance what is enough retrieved dropout. And actually, I would be more in favor to clearly pre-specify and don't allow any choices, and then you can still do this as a sensitivity analysis. But
I mean, to comment to that, all these questions are, of course, very unconcrete, so they are quite vague in some sense. And here you assume, at least I think that's what you do, that these are not terminal events, what you have, like, for example, death are not part of the intercurrent event list, which is hardly ever the case. And then what you also do is you assume them all to be of the same meaning for patients, because you simply propose one approach to handle these intercurrent events and the missing values after these intercurrent events. And I would again think that one important step is to try to understand what is the nature of the intercurrent events, and to also do simple statistics on just listing these intercurrent events by treatment groups might help a lot to understand whether they are related to the treatment or not. And this might also impact your choice of estimate, and which is sensible in the end. And then if we are there, then there's different questions. I think from a regulatory perspective, sometimes we are interested in conservative estimates, because these somewhat protect us from making wrong conclusions. But then from a clinical perspective, of course, we are also interested in unbiased estimates. And this is like a different question. So for the SMPC, maybe an unbiased estimate might be more important than for the decision-making process. So that's also why we usually see more than one estimate, or more than one estimate for the estimate. And this is maybe, so it's a bit hard to answer these questions because they are maybe too simplistic.
Yeah, maybe just to clarify, we are in a setting where we have a non-terminal intercurrent event, and we assume clinically it's plausible to use treatment policy approach for all intercurrent events.
I think I'm more or less echoing what people have been saying. I mean, from the discussion at CHMP and the text to the SMPC and so on, it's kind of obvious it needs to be clinically plausible. If not clinically plausible, then it doesn't really make sense, even if it's statistically okay to do it. So I would say that is sort of the starting point, but not making things easy, of course. And in my perspective, I would think I would be most worried about bias. Biased estimates is definitely a problem. And depending, of course, I can live with some conservative bias, but not the opposite. So it's difficult to have to give a general view, but I think that would be my sort of easy perspective on this one. And following up on what Kit said, we see a lot of trials, unfortunately, that where patients are not followed up after the intercurrent event, even if it is not death. And that destroys the trial oftentimes. I mean, if you do the follow-up and actually follow up until the endpoint, whether a intercurrent event has happened or not, you will have a lot of information. And what CHMP will oftentimes ask for are, of course, sensitivity analysis. First of all, a justification for why you did it the way you did, and then if you are not really happy about the justification, or if we believe maybe we don't really understand what's actually happening, we would ask for sensitivity analysis. So that is always an option to sort of list, this is our primary one, and we will do these different types of sensitivity analysis.
Yeah, thank you. I mean, I think you mentioned a very important point about a clinical possibility. Just from experience, we've sometimes seen cases where having a conservative bias was prioritized over the clinical sensibility of the imputation method.
By regulators or by...
Maybe that's what I meant. For the decision-making process, it might be that we might be more willing to accept a conservative bias rather than an unbiased estimate. But then for the estimation of the treatment effect, it might be different. But of course, that complicates matters. But we have different estimates for different purposes. Maybe that's also what we need to acknowledge.
Right. But it's the same estimate, but different estimates, right?
Maybe it's even not that, is it? I mean, it depends a bit, but you try to estimate the same estimate. Yeah.
Karisha?
I think The main consideration really is to try and minimize the problem and the importance of pre-specification. You need to pre-specify an analysis of a choice that is well justified from a clinical perspective. And regulator usually, when they see that, if in light of your data, if there are some concern, they will always request additional sensitivity analysis. So in terms of satisfying ourselves with the fact that the estimate of treatment effect is unbiased and is robust, we are at liberty to ask for additional analysis at the time of assessment. But the important part from your side is to have clearly everything justified and laid out and pre-specified, because if it is not pre-specified, then it becomes a post hoc analysis, and that is more problematic.
Thank you.
Last comment from Frank.
Yeah, for the first question, I think the understanding of the clinical setting is extremely important. What would happen after treatment discontinuation, either after taking placebo or after taking the treatment, would need to be known. So would there be a slow decline, a steep decline, a relapse? Would it be different between the two treatment arms? That helps you decide on which imputation method I think you want to choose. And then I think the statistical properties come after. I think the clinical setting and the impact of what happens if you stop treatment comes first. And then, yeah, on the second one, I would have said type 1 error first because it helps decision-making. But then we heard what Eva said in terms of information in the SMPC. Maybe I would like to add that maybe it also depends how many trials there are that look at these questions. You know, are there several pivotal trials? Do you have phase 2 trials as well? Maybe you have already a lot of, or you have other information that you can piggyback on to help inform the SMPC. So that can play a role as well.
I think we need to move on.
Okay.
We have a whole session on estimates this afternoon, so I think we have time to discuss there again. Thank you, Tobias. So thank you, Tobias, and then next one is Frederick. So now we change to something else. Okay, Lawrence?
So thank you for this opportunity to present. I'm Frederick Earn from J&J. So this is a bit different topic than the previous ones. So I want to talk about very often across our programs, we still, rather than conducting a single pivotal trial, see.
She said it will come. Let's trust Lawrence.
Yeah, it's good. Yeah, so basically the two trials rule, I think you're all very familiar with that. Very often we will have a single primary analysis in our trial, in our confirmatory part of the program. We'll have done two, sometimes what you might refer to twin trials. There are essentially no obvious difference between these two trials. We might then have a one-sided test versus 2.5% for each of the trial. We will sort of declare this as a positive program. If both these trials are positive of the primary endpoint, there are of course many other aspects that are needed for approval in terms of benefit risk and so forth, but this is still seen as maybe a prerequisite. I think there are exceptions. Oncology, I think it's more common not to do this. Rare diseases, of course, I would add large cardiovascular trials. I think I'm sure there are other exceptions as well, but still, at least in my experience in the therapeutic areas I worked in or working in currently, it's very, very common that we mandate that the two twin trials are essentially mandated. So you could think, could there be alternatives to this? Well, one thing that has been written on, so this is of course not totally new, this question. You have a single trial and you might then say, I'm gonna test now the primary null versus 2.5% squared in terms if you want to have the same risk of approving a non-efficacious drug, so 1 over 1,600, you could debate, and people do, what should these numbers be? But in terms of here, if I want to match it and say for the pool trials rule, I want to have the same risk of approving a non-efficacious drug under the null, you could argue for this level. And people have looked at this and shown that actually there can be Stephen Sen quite some years ago now, and others have looked at this and shown to at least some power gains in this setting. So I've just listed here some basic pros and cons that I can see, some moderate power gains, but I think there are also other, just in terms of something, you have something as simple as a futility analysis. I think most people would then say, we should, of course, account for both trials in the futility analysis, but already then you lose the independence. And you can, of course, imagine many other complex adaptations or just early stopping, etc., where it also will be, I think, advantageous to have a single trial. For sure, and I mean, at the end of the day, interested to hear what regulators would say, but I think you need to make one decision, and it then makes sense to me to synthesize all this information into a single analysis at the end of the day, anyway, if these are twin trials. Of course, you could see it is just a route to make smaller programs that will not necessarily be advantageous. Also, I mean, sample size is not necessarily only driven by efficacy, of course. We need to assess benefit, risk, and safety, and so forth. But I think that could be mitigated by just keeping the sample size the same, take any gains out on the power side. And of course, some people are concerned about this, the final bullet here, the independent replication. So I think my kind of modest proposals, I think there have been other proposals to say we should use phase 2 data, we should model mechanistic models, etc., replace one trial. But here I'm not talking about really replacing any data. I'm just saying, could we do, if we do 2 trials, each with sample size n, if that is deemed adequate to support approval, well, what about one trial with sample size 2n? And then you could debate the alpha level, but let's say one side the alpha square for the sake of argument. In which situations could 2 replace 1 here?
Thank you. I think that's a clear, very as fundamental question as it gets. Khadija, start.
Yeah, thank you. So, sorry, I figure out what to do. So that's really an interesting question. I think there is no regulatory requirement that you must do 2 trials, but of course, if you do a single trial, then the evidence needs to be both compelling from a clinical and statistical perspective. Now, with regards to why do we ask for 2 trials? So the advantage is that you want to replicate the evidence. But we need to understand what we mean by replication. If you are just going to conduct study 1 and study 2 from the same sponsor, is it really a replication? Because the advantage of replication is that when you move away, say you do your trial, somebody else independently does the trial produces the same result. That gives you more confidence in not just the efficacy, but also the effectiveness of the trial, very much closer to what would happen in real life. So in that situation, if you are just going to do study 1 and study 2, then of course, my personal preference would be a 2N trial, because then you will get much more precise estimate of your treatment effect. But then if you have the opportunity to do 2 independent trials in a different setting, then maybe 2 different trials are, important. Again, it depends. So that's my take. Thank you.
It's the safe answer always. It depends. So Lucas, on what does it depend?
It certainly depends on a great deal of things. I think that Khadija raised a point that is very important for this discussion, and that is, what do we need the replication actually for? Because replication is maybe not a value per se, but we can use it as a line of argument. And also, one thing that I have observed in applications with single pivotal trials over the last years is that when you run into some sort of signal from subgroups, for example, it's going to be very hard to discuss whether and to decide whether the signal, if it's like of a size that it matters for benefit risk, whether that signal is credible, whether it's plausible or not. And then the tools that we have for this is clinical plausibility or biological plausibility and replication. So if you have a second trial that maybe does not need to be identical in its design, but if you have a second trial that replicates the subgroup finding or maybe does not replicate the subgroup finding, then the discussion gets a lot easier. So this is really, for me, one argument why I, in most cases or in many cases, prefer to have more than one pivotal trial. But then you could also make the argument that if the study is large enough, you can have some sort of internal replication. And I think this is discussed with multi-regional trials, where you consider different regions as a replication. So I would say it depends, but two trials can really safeguard you from a very complex discussion of signals that might come from somewhere in the trial.
Benjamin?
Just a brief comment. If you have two trials, you can still combine these in the estimation process. And there are situations where, for example, we have some endpoints where the power is so high that we can do separate trials and we can have two trials, but there's endpoints such as mortality, which is observed only in rare cases. And then it's foreseen already in the trial plan that these will be analyzed on a pooled manner. If we have rather high power, I don't think that the power gain of a combined trial is of such high importance, especially if we want to keep the sample size fixed. It is more of an importance when we have low power, and then we already now pooled trials. And I think this also somewhat then, together with what Lucas said, I would have a rather strong preference for two trials rather than one. And I think the operational hurdles with respect to, for example, interim analysis and so on, they can be overcome by what Kasper called good, how did you call it? Good developers. And I think maybe that's what we need to agree to. And then again, it depends. Do you have twin trials, they run in parallel, or do you run them in sequence? Then maybe this problem is gone anyway. So, but in principle, I would give preference to two trials. But as Khadija has mentioned, there's no strict rule in requiring to have two trials. So you can run one trial, but with the caveats Lucas has mentioned.
Catherine?
Okay, don't forget to turn it on. My preference is also for two trials, mainly because of bias considerations. So if it must be more likely to accidentally observe or have bias in one trial than accidentally have some bias in two trials in the same direction even. But then again, as Katja mentions, if the trials are identical at the same sites, are there really two trials? Yeah. Regarding in which situations it could be very beneficial to have one large trial, for example, for urgent vaccines or cases like that. An example that immediately comes to mind where it could be very beneficial.
Thanks. Actually, there was a whole session in the CEN last year in this place on replication. And I want to take a bit of a bigger picture view on this question, in the sense that we had a substantial crisis in science, not only the medical one. And that was very much driven by the concern that lots of the research that's published, that's out there, is actually not reproduced, not replicated. So replication for me is a cornerstone of doing a scientifically sound job. It's not to achieve a significance level of 0.05 squared. That has actually nothing to do with replication. So replication itself is a value we should actually cherish also as a statistical community. And it's actually a pharmaceutical company among them expressing their concerns, Amgen, on the early work done by academics, that they had problems in replicating earlier experiments. And I think we should constantly be aware that this is the reason for replication, not achieving a squared of an evidence level. And there is guidance, because essentially the meta-analysis is one pivotal guidance. If I would summarize why it's there, it's what's needed to consider actually foregoing replication. So implicitly, that guideline already is quite clear that we expect some kind of replication in a clinical drug development program, and there are conditions to fulfill if you want to deviate from that. And it's also not a coincidence that the 0.05 squared is not mentioned there.
Could I also ask on that? Could a replication, though, be from rather phase 2 to the confirmatory phase rather than?
That's not replication, because the phase 2 is there before you decide to go into phase 3. If you think confirmatory, replication is is replicating the confirmatory experiment. But it doesn't, I would very much prefer it's not the same type of trial. So the twin trials also give very little additional information.
Frank?
Yeah, I was going to mention the reflection paper on one pivotal trial and meta-analysis. There are eight points to look at, and only one. So one of them is the statistical significance. So there's a lot more to do. And we've seen an example in recent years where there was one pivotal trial. We did not understand very well the results between the two doses, the two treatment effects. And my experience is that when we arrive at this stage, there is a tendency for all stakeholders to be biased towards the alternative. We want to believe that it can work, especially in a disease where there are no treatments or very few treatments. And then we're biased towards approving the medicine. And then, sometimes then we see that we've made a mistake. And that's why you see the need for replication. And I think if we read E17 on multi-regional clinical trials, it also gives some clues about one of the aspects. So looking at subgroups, how it was mentioned before, but this is only one of the many topics we need to look at. I think maybe the question would have been two similar trials or two same trials versus two slightly different trials. I think that's the question, maybe for next year.
Yeah, I can make a provocative question for next year. One trial with 2 N patients, but 2 subgroup analysis as primary analysis for replication.
Okay, comments from the audience? We take one comment and then we move on. Do you have a mic?
What's the favorite answer of a statistician? It depends. What's the favorite answer of a regulator? It depends. Now you have a panel of statistical regulators.
N squared.
Now it is. Pierre Verwij from Idorsia in the Basel area. Can I ask one question to the panel? Before the break, we discussed speed. And I would say that in this setting, if you do a single trial with 2 N subjects, that would finish earlier than 2 separate trials with N subjects, even if they start at the same time. Of course, there's not a reproducibility that you can test here, but you can gain some speed, which, according to the session before the break, could be advantageous. Is there an opinion about that from the group?
Yeah. That's the microphone, please.
So I think this question is not about speed at all. Speed is about what is the minimum you need to do to get approval, to convince regulator to approve your medicine, regardless of the level of evidence. And it can be low, it can be high if it's better. But if it's low and it's low and high enough, then that's it. You've won the speed argument. And here, what we're talking about is level of evidence in general. So we're talking about the first element in the first session, which was level of evidence or something like this. So 2 different questions.
Yeah, Pierre Mancini, Sanofi. I'm wondering, I mean, if there are, in your view, some other possible avenues, which would be to do a robust phase 3 trial. So that is at the end significant. But then the second, or the replication would come to a lighter phase 3 trial. For example, a pragmatic trial that is, leveraging real-world data, for instance, in the endpoint versus a central assessment. Or even, I mean, some real-world studies based on, when the drug is on the market. So replicating later, I mean, maybe having a conditional approval. So, and, would you see any kind of disease where it would be more acceptable than other?
Anybody?
I think the question is for Eva because she presented about conditional marketing authorization.
Thank you. I think there would be instances where we could have this sort of proper phase 3 trial, sort of the ideal one, and then replication in a sort of a lighter setting. I think that would be possible. So as for me, and I think for many CHMP members, but I will speak for myself, I think the replication of some sort is the key. And it's even better if the trials are in some sense independent, because that gives you some kind of comfort that this is not really a specific thing that might be biased, because we see the same thing in a different setting. So probably reluctant yes to the first question on a kind of a pragmatic trial as the second one. I would be more worried about using observational data as the replication. I wouldn't regard that as a replication. It would be supportive, but that is a different thing. Yeah.
Sorry. I didn't want to interrupt you. Okay. One point regarding the pragmatic trial, after you have got your license, often case it's difficult to run it. You can go to another region. That might be one way to do it, but oftentimes I think it will be very hard to recruit patients to such a trial if you have shown clear benefit over the comparator drug. And then we lose that kind of additional evidence. In principle, I would completely agree with you. It would be good to also have this pragmatic trial to understand how the drug works in a more realistic scenario with less rigid inclusion and exclusion criteria, and perhaps also with a lighter weight on the measuring of the endpoints. But that's maybe a different question. And the second point why I think it's usually not feasible is during discussion for the CMA or MAA, so for your license, we don't have the replication at hand. So the discussion Lucas mentioned, we see a subgroup result and we don't know what to do with it. We have that problem during the assessment, and we cannot say, well, we postpone that discussion until very much later. So if if the signal is strong enough that it might pose a some risk, or it might increase the benefit in a subgroup to an extent which we believe that the effect is driven by that subgroup, that might impact your label immediately. And that's also a risk you need to take as a developer. So we might restrict your label in that case. We might need to.
Quick comment and then we move on.
Yeah, just a quick one. I was assuming that this pragmatic trial would be before licensing, just to make that clear.
Yeah. Okay, great. Thank you. Fredrik. Then we move to a second fundamental question on multiplicity study design. What are we actually trying to answer? Yes or this?
Like this? No, that doesn't seem right. I think it's
Okay.
this way, isn't it? No, that doesn't seem right. Oh dear, this is tough. Like this? No?
No, I think you need to turn it the other way around.
Very complicated.
Yeah.
This way.
This way,
Okay.
that way, yeah. And then it's on your ears.
Statisticians, you know,
Yes.
we're not good with these things.
And you can just hold this
Okay,
one.
can you hear me? Yes, I think it's working. Thank you very much for the invitation to present this case study. I must say I enjoyed this session enormously because, you know, it's very rare to be able to discuss some topics like the one I'm going to discuss with regulators, and have their feedback. This is this question I have here is about a trial that I was involved in a few years back. I was on the IDMC of a trial, and I've tried to kind of blind the details of the trial so that I don't offend anyone in the room, if people are familiar with this trial. And so I've tried to anonymize it to the extent I can. So this is a trial in advanced cancer, and there were 2 experimental arms tested in this trial against a control. So similar to the trial that we've seen this morning with Casper Ruffibach, but except the control group was a real control, and there were 2 versions, if you like, of a new treatment. And there are many situations where we have this. It could be 2 doses, could be 2 scheduled. In this case, it was just 2 experimental arms, but one of the 2 was the preferred. They call it the preferred experimental. So they were really hoping that the arm A would be the best arm and that it would be approvable. There were not only 3 arms, so multiple comparisons there, but also 2 outcomes, as is often the case in advanced cancer, PFS, which was the primary, and OS, which is the secondary. So sort of typical situation. And this is the testing procedure so I've extracted this from the protocol of the study, and this was the testing procedure they used. So if you look at the graph, it's pretty clear. They tested PFS for A versus C. Remember, A is the preferred experimental, C is control, and they tested that at 5% level. If that was significant, then they would go down to PFS of B versus C at the 4% level and OS at the 1% level of A versus C, again, the preferred arm. But of course, if PFS of B versus C was significant, they could call back the alpha level of that comparison and test then the OS at 5% as well. And then if that succeeded, then they would go down to the OS of B versus C. Now, back then I sort of disliked this, intensely. I thought that this was just not right. And I sort of forgot about this. And then recently I was, looking at a guidance on patient centricity in trials. And I was wondering, how the heck can you explain that testing procedure to a patient? It just doesn't make sense, right? You can't explain it. If you don't have a statistical degree, it just doesn't make sense. So the issues here, in my view, are that you test OS only if PFS is significant. And we've seen, and you regulators have seen a number of cases where OS was actually quite convincing, but PFS was the primary outcome and you couldn't test OS. So that's a problem. And the second problem here, which is specific to this multiple comparison, is that the OS of A versus C, which remember is the preferred arm, is only tested at 5% if the PFS of the other arm is also significant. Now, surely that doesn't make sense, right? So I would suggest that there is an alternative way, which is probably something that didn't exist back then, but it exists today. You could use an analysis of generalized pairwise comparisons, and the win ratio is the most commonly used form of it, unfortunately, because it's not very good. But the win ratio you can use, and then use an analysis of time to worst outcome rather than time to first outcome. So you look at time to death first in your pairwise comparisons, and if the patients aren't dead yet, you look at time to progression, which makes a lot more sense than looking at PFS and OS using this multiple comparison business. And then if you do that, then you have one analysis for each of the 2 experimental treatments. And of course, you have to adjust that. There's no way you can avoid adjusting that comparison because you essentially have 2 questions there. So my 2 questions would be, first of all, am I right in saying that this is really hard to explain and perhaps even acceptable by agencies? I don't know that. It probably was accepted back then. And the second question is, would you prefer to see an analysis like the one I suggest here?
Thank you. Very nice and clear question. I think the first one is about the something is statistically correct. Does it make sense, basically? So who wants to go first? Brian?
Yeah, thank you. I'm not so sure that the other procedure is so hard to explain, but I'm biased. I've worked on these kind of procedures before. I mean, it's just a way of distributing your chances in getting a claim on one of these endpoints. And talking about the other one is you you propose a completely different endpoint, and now we are missing a clinician on the
Yes,
panel.
we are. We
to
are.
tell us whether an endpoint time to worst event is something they would trade for in making the decision in the end. And I think it doesn't really resolve the problem, because in the end, you may still want to know about overall survival for both drugs, or even the effect on progression. And then if you want to derive claims on them, you would again have to put them into a more complicated multiple testing procedure. And maybe a final point here is that since you have a preference on A, if you use this approach and then just use a Holm there, it may not be actually more powerful, because that just means you do away with your prioritization of drug A.
Sure, no, you're right. You kind of forget the fact that one of them is preferred.
But you could do a hierarchical test, and
but you know, you
or
always
two other
have the
weights and
two-sided coin. If you prefer one test, you do it first, and then sequentially you do the other, but you might run a risk of then, you know, ignoring the fact that the other treatment was actually better than the one you preferred. So, I mean, it's always a question about sequential versus another form of adjustment.
You can choose weights in these procedures.
Yeah,
as
you could
see
use.
fit, and that's more or less like, it's a way of allocating your resources to answer the questions that you want to answer in the end, because you can only answer a finite number of questions.
No, I agree with you fully. I mean, the problem is that we only have statisticians here mostly, and not clinicians, and more importantly, not patients. But I mean, look, you can't make sense of this graph here to someone who doesn't know statistics. It just doesn't make, I'm sorry to say, it doesn't make sense. I've tried it and failed. Maybe I didn't try hard enough, but it's hard. I mean, it's a hard way of splitting your alpha level, in my view, and it doesn't make sense clinically or for a patient.
Yeah, but I think we have a problem. Do we really want to choose our endpoints in dependency what the patient can understand or not? I think this is one point. And I think the second one is also what Florian, the point Florian made, does it really make sense to change your endpoint because you have a statistical problem? Of course, then I mean, it's really
Yes, it does. Yes, it does. Yes, it does. I mean, I don't know how many times you've discussed as regulators, should I look at PFS first or OS first? I do consulting for drug companies, and that's the number one question. Should we use PFS or OS? And to be honest, you know, it's a real question. It's not a statistical one. It's a clinical question. In my opinion, it's more of a clinical question.
But my main problem with this is that I think it's really difficult to compare this endpoint between trials, because, I mean, it depends on the distribution of follow-up of patients, what the endpoint is. I mean, indefinitely, it will be OS, because indefinitely all patients will die sooner or later.
But that's the problem you have with all analysis types, not just the one that I
No, but I mean, if you have a trial with a very long follow-up of patients, it's OS. If you have a trial with a shorter follow-up, it's PFS. So it's really difficult to compare this, and I don't think this is really a good solution compared to OS and PFS, where you can hope that you can make this comparison between trials independently of follow-up times.
If you assume proportional hazards, you can, yes.
Yeah.
That's an assumption.
Yeah, of course.
Starting from a different direction, to start with your point on the let's say the redistribution of alpha scheme. And I think, to be totally honest, I kind of share that I see schemes that are indeed correct in terms of controlling the time on error, but where I have a difficulty to see the full logic there, and whether I think it's actually consistent with what's in terms of what are your priorities and estimation and knowledge that you would like to gain from the trial. But they still might work out that way. So you can't immediately see from the scheme how it actually behaves in a practical situation. So I kind of share the not being entirely comfortable with schemes I can't fully grasp in terms of what's the real strategy behind this, and here you're clear. So I think there's work to do there for us as statisticians, but also for us as regulators in the communication we have. I think, yeah, we are discussing about changing endpoints, but it's proposing a different design, essentially also a different research question and a different estimate. So which I think could be considered, but all of that's happening. So you indeed lose somewhat of the landmark against existing trials, existing treatments, and I think that's also the way that Andreas. So your reflections there would be useful. The other part is, it doesn't stop with the hypothesis testing. The information that's in the trial is in the trial. You have information on progression-free survival. You have information of overall. So how do you think that should enter the design question? Also reassuring Andreas's question, because in the end, we indeed might need more information than just the outcome of the first comparison to be made on event-free survival.
Yeah, I mean, again, if you take the patient point of view, it's okay to do multiplicity adjustment if you have more than one treatment, that's for sure. No other way to do it. But in terms of endpoints, I just think we miss what's important to the patient, which is that all endpoints are important to them in some sense, you know. And when you look at them separately, then we have a hard time recombining them in a sense. What I propose is to have a combined endpoint that is patient relevant.
Please.
I think that's a valid point. I think the issue is how do you communicate that to the patient? I think when you first started, you said, you know, it's difficult to explain this type 1 error control to the patient, but even more complex when you have this way of putting the endpoints. Also, how would you communicate? How would you communicate that to the patient?
That's another topic, I think, but it's a valid one. I think it's an important. There is a session on patient preferences, right? Maybe we'll touch on it tomorrow. That's a really hard question, but have you tried to explain a hazard ratio to a patient?
Yeah, but I mean, I was just making the point that, you know, if you're thinking about communicating with the patient, of course, you know, for regulatory decision-making, type 1 error is important, but also when we look at PFS and OS, we look at them in combination as well, because we need both to make a decision. So if you have that data, you have to consider both. If you are now moving towards a time to an endpoint in order of priority, that may also be an important one. But I think what I'm trying to do is to make the same argument. If you know your motivation is communication of
right,
then
take this example. And suppose PFS is non-significant, and the OS shows a really, you know, very positive result in favor. How do you explain to a patient that that doesn't matter you because PFS is non-significant? You can't claim the OS benefit. That's the way they test it here.
Yeah.
So does that make sense?
No, I mean I can see the dilemma, but I don't know if you want to.
Maybe not that question. I think my problem with the proposal is we are combining multiple questions and changing multiple things at the same time.
Sure.
That makes it quite hard.
Let's change it, yes.
For me, the question about how do I explain this multiplicity graph to a patient is maybe not of such high relevance. It's a trial design feature. We don't explain all the trial design aspects. We don't explain all the endpoints we measure in a meaningful way for patients. I believe that there needs to be expertise which goes beyond the expertise a standard patient has, but in principle, the trial should be understood by the patient. That's a different pair of shoes than explaining all the details of multiplicity. I think it's even the question of what does multiplicity mean and what is multiple testing and what is type 1 error is completely out of scope for many, if not most patients.
I know, I agree.
So the question of whether this makes sense maybe is a question we need to discuss with the companies, and I know why this graph happened as it happened. PFS is an earlier endpoint. You try to increase your chance of winning. That's why you put it first. And then you want to go to OS because you also need to have OS to make a strong claim. And that's why it happens. So there's also a rationale behind this graph, which I do understand. The question is, why don't I recycle, for example, the alpha? Well, that's another one. So I could increase the chances of winning. The question of why do I test PFS, OS in the way it is put there, and why do I give preference to the other treatment, to one of the treatments, and why do I put it in the graph like this? Maybe it's a good question to be asked, but these are questions which could also be solved by your approach. So we could use Holm in that graph as well. So we could have PFS and OS in the two trials, and then we could split the alpha with using an approach based on Holm. So this would all be doable. So this is the one part of the story. The other one is the question of changing the endpoint. And this is maybe more complicated, but for me, I'm still not convinced, so I have to go in the same direction as others, that we really gain by having this composite endpoint as to what we have now. We want, I believe, even if hazard ratios are complicated to be explained to patients, we also have other tools of explaining overall survival data. For example, we have Kaplan-Meier curves, or we have landmark survival. So these are tools which are more understandable. I do understand my percentage of the probabilities that I will survive a specific amount of time is, I do understand. I believe every patient does understand this, at least roughly, maybe not correctly, but roughly. And this gets lost when we only look at this endpoint. This is one way of maybe improving your testing, but then interpreting the results and making them accessible to patients requires additional work as well, as for all endpoints, by the way. So I'm not sure whether it solves so that's maybe the difficulty I have. Do we want to support patients? And I'm not sure whether this supports patients. Do we want to support drug development? Maybe it does. But I think we solve some problems, we get other problems. So the question is, how do we want to weigh that?
You, Eva? No.
Yeah, I was wanting to say something like, in the end, we want some estimates on overall survival. I mean, that is what we understand and what really matters to patients.
Yeah.
Yeah.
The first analysis they would give you that.
But so in some sense, the other one is kind of a failed trial design, because you have some kind of belief that you should quickly be able to show efficacy on not time to progression, but progression-free survival. And then it fails because you haven't really understand the drug mechanism properly. That is the way I'm thinking about it. So in terms of wanting speed, you take a too large risk, and then the trial fails. And that is sort of another way of seeing the whole thing. Maybe those kinds of spending alphas and in complicated ways are not very useful, because there are some risks there.
I don't disagree with you. You know, this analysis looks at all deaths, so it does look at survival as it should. And for patients who are still alive at the time of the analysis, you then take their time to progression. So in sense, you use the best of both.
The one I was criticizing was the other one.
Yeah. Oh, sure. Oh, sorry. Yeah. This I think addresses exactly what you point out. With the problem that I agree with is that your analysis depends on the follow-up time. That's the limitation of this approach for sure. It changes over time. The estimate is now dynamic. It changes with the follow-up, which may or may not be acceptable.
Yeah, I think my main message was a general one. Don't destroy the opportunity to actually show something on overall survival.
Absolutely. Yeah.
Last comment from Florian, and then we need to move on.
Yeah, and I mean, you can do this with a graphical approach. You just have to put other weights in there. Maybe a last argument regarding explainability. If you propose a Holm here, you can also paint the picture for the Holm procedure, and where you also have this problem, you test the one treatment at alpha half, and only if you meet that, you test the other at 5%.
Yeah.
So that's the same thing, difficulty to explain it to a patient. So it's just, I think, not everything in statistics has to be explainable to a patient.
I have to have an argument, right? So yeah,
I fly in an airplane and I don't know how it works.
Yeah,
so
agreed.
Thank you very much, Mark. Good discussion. And then we have third fundamental question from Kostas on exploratory analysis.
I don't know how to put it. It's like this.
Yeah.
Can you hear me well?
Yeah.
Cool.
Yeah, my name is Kostas. I'm data scientist from Novartis, and we put together with my colleagues Mark and Bjorn this question. And the plan was to have some animations in the slides, so not to overwhelm you with the text. This will not work, so bear with me. You'll see a lot of text, but the message is clear and simple, I hope. So the question that we would like to ask, I told you, bear with me. The question that we would like to ask is, what are the quality standards for exploratory analysis from a regulatory perspective? And let me unfold the thinking process behind this question. And we think that it's very critical to have the statistical thinking. It's a very important tool for us to ensure some quality standards. Also, it's the fundamental tool to answer questions with the data, get insights from the data. But if we want to get clarity, reliability, and replicability in data analysis and the decision making, the statistical thinking should be combined with some structured frameworks. And this is another attribute of quality that we want to emphasize. And there is plenty of frameworks out there. One effective and general framework that embodies statistical thinking is the PPDAC, which stands for Problem, Plan, Data Analysis, and Conclusions. And to the top right, we have one PPDAC cycle that is called. And this framework provides some structured approach for problem solving by guiding the researcher through each steps. And I think this is very critical. And let me outline briefly the steps. In the problem phase, we need to outline the question that we want to focus on. And of course, identify what is the quantity of interest that we have, what is our estimate. And this should be aligned with the question. In the planning phase, we have the data and analysis strategy, again, aligned with the question, be fit for purpose. In the data, we have the data collection or some initial data analysis that needs to take place to prepare the data in some form that we can use later for the analysis, which comes in the fourth step. The analysis can be statistical, machine learning, whatever type of analysis fit for purpose for the question that we have. And of course, some desirable characteristic is to be reproducible and accurate. And finally, we have the conclusions or the communication, which again should reflect, always keep in mind what was the question that we started for, and should be clear and transparent in the reporting. And the PPDAC cycle, it's very versatile. It can be used to answer both exploratory and confirmatory questions. And this audience is more familiarized with the confirmatory analysis, and we have the primary analysis of the clinical trials and all of this estimate framework. And you can see that one application of the PPDAC to answer questions around causal questions around the treatment efficacy. But of course, we can have it also for exploratory analysis. One attempt that we did the last period to assess treatment effect heterogeneity, and we introduced some workflow that it's some sort of exemplifies the PPDAC to this question. Now, why we are asking this question and why we are bothering about exploratory analysis, we think that it's very crucial to answer questions of interest for drug development. And many times it influences decisions of approval or also, of course, internal decisions. So following some quality standards and being transparent in this type of exploratory analysis for us is very critical. And here is some example analysis that I'm more familiarized. I'm sure there are many more. We can perform some exploratory analysis to identify prognostic variables or create with machine learning super covariates to use later in some trials for covariate adjustment. We can have assessments around treatment heterogeneity and, of course, safety assessments. And many times the exploratory analysis is a bit undervalued, but in my mind gives this flexibility that we need to learn from the data. And of course, this flexibility comes with a price. For that reason, in this type of analysis, we need to be self-disciplined and rigor and to ensure some high-quality outcomes and outcomes that we can trust that can be replicated. So while there are very well-established standards for primary analysis, guidelines for exploratory analysis are less clear. Here are some examples from some other areas. In pharmacometrics, we have this model-informed drug development, which they suggest a structured framework for credibility assessment. And there is some other document about the good machine learning practices for medical device, which they suggest 10 principles to sort of, provide some guarantees of high quality device. So with this context in mind, we would like to open the discussion about the quality standards of the exploratory analysis.
Thank you. Introduction. Adisha, you want to go first?
Thank you. So I think the first thing with regards to the quality standard for exploratory analysis is really stating clearly the objective of your study. And that goes with whether it's a confirmatory, exploratory, it doesn't matter. You need to be clear about your study objectives, design your analysis, and collect the data to be able to answer the key questions of interest. And I think that there are many guidances with regards to data quality that you should follow. For example, in terms of having an audit trail, etc., compliance with GCP principles. Again, there are other considerations like statistical aspects with regards to machine learning and so on. There are other guidances there that are available, but really the building block is really being clear about your study objectives, setting those clearly, have a pre-plan and have multidisciplinary discussions. If we are targeting a particular estimate, have those discussions early. And yeah, so that's my input. Thank you.
Yes.
Somewhat briefly, but I do acknowledge, and I think I also very much appreciate highlighting this, because I agree with you that it doesn't stop at confirmatory analysis. You could even wonder whether, in terms of the assessment of data, even at the confirmatory stage, is also looking at lots of analysis we would call exploratory. So I think it's well recognized, and it's also recognized that we don't have standards that you can easily point to. What I would like to promote here, which you're mentioning the model-informed drug development and the credibility framework. I'm assuming you're aware that credibility framework was published by the Modeling and Simulation Working Party. So that has actually a regulatory basis. So it's a very, very good starting point also for exploratory analysis. I would probably not think of another framework. I would actually really start there because it's quite comprehensive and has both a research question and regulatory risk.
They're too close the microphones probably.
Yeah. So it has a very much research question driven, which I think is what Khadija already clearly indicated, as well as regulatory risk or risk for your decision-making entrance. So I would really promote to take that as a starting point, but we don't have standards at the moment, but I think it will be included. So that's the discussion ongoing. You're aware of the ICH guideline on developing this framework will one way or other find its way into that ICH guidance. And I think it's a really good starting point also for this discussion. So I would rather promote not coming up with something entirely new.
I think everybody should know, if they don't know what Kostas was talking about with this MIDD framework, to do some homework. Yeah.
Yeah, we have also the references in the slides, so we can find. Yes.
Also enough references
even
for 2 minutes.
Yes, sorry.
For me, it's not so much for regulators, but for public health and for wasted research. So Kit said earlier that a lot of drug development is continued, even though there's little chance to prove that there is a positive benefit risk. So if we do a better, so if we are more strict earlier in drug development and with exploratory analysis, then we will stop developments that will probably lead nowhere and focus on the ones that are more likely to succeed. And when I was in industry, I did a lot of bad exploratory analysis, and the clinicians were believing in it. So.
Yeah, I guess this was the starting point also in our thought process. And, when we say quality standards for regulators, the quality standards should be across. If you have this quality framework, you should use it as a sponsor regulator or public health, of course, yeah.
Lucas,
yeah, basically not much to add because I think that general appreciation was already phrased. I think we can, there's a lot of room to improve for exploratory analysis, also in the regulatory context. You mentioned safety, for example, exploration of safety. And in that instance, I remembered something that I recently saw with a company that made an application, had a quite complex framework for their exploratory safety analysis. And basically they used it as a screen to screen for those adverse events they wanted to include in the SMPC. And I thought this is a great way to do a first screening. It really helps kind of to filter out those events that don't need any discussion because you see a clear signal. But then for all those that were not selected by this, we had to have a discussion nonetheless. So while I think that there is room for improvement, definitely, I don't believe that this will like make all the discussions that we're having now on exploratory analysis vanish. We still need to discuss a lot. Obviously, that's the nature of exploratory analysis. Yeah. So key message, I think, from what I wanted to say is that this is great and we have room to improve.
Thanks a lot.
Any comments from the audience? Yeah.
Thank you. That was fascinating. Just a question to the panel on what is an exploratory analysis, I suppose. And so in terms of the examples you gave, two of them are more exploratory than the first one, in my opinion. But I don't know, when you're talking about using a super covariate, but presumably in the primary analysis model.
Yeah.
So isn't that
you use some exploratory and then influence the primary analysis. You know, you identify something, you build a lot of models, you had some strategy, but I don't know if this can be qualified.
But what I meant is I would imagine the regulatory standards for that are high, as for the others, they are exploring. So that it's a sort of slightly different thing, and that might be useful to define what you really mean by exploratory.
I can just echo what you just said. I think for me, it was also a bit difficult to understand. And sometimes, I mean, the difference between purely exploratory and secondary and primary, well, it's a continuum. It's not a strict rule where we can just make a clear cut. The more exploratory you get, and that's maybe also the framework tells us that the less rigor we need, the more relevant it is for the information on key features of your product, or possibly relevant, the more important it is that how you pre-specify and what you pre-specify and how much rigor you put into your analyses. And I think that is, I also think it's good you bring it on the table, that we also understand, everyone in the room understands that it's not, we don't only take care of confirmatory analysis, and it's not only about the p-value. And I think we heard it repeatedly over the day. Eva has mentioned it in her talk. It's not only about the p-value. Yes, multiplicity and p-values and all that is important, but there's much more to it. It's about bias, and it's also about the totality of evidence, as we call it. So yes, we look into multiple endpoints, and the question is, what role does this endpoint play? And if it's a biomarker endpoint, well, then it more or less immediately it becomes from an exploratory endpoint to a key endpoint, and then we have the in vitro diagnostic regulation in place, and you need to do much more for companion diagnostics and so on. So then you see it changes, that the role of this endpoint changes during your development process. And maybe this should also be incorporated in the development plan, not only for a single trial, but for the range of trials you plan. And I think it's a good way to put this on the agenda here today. Thank you.
Yeah, thanks.
I think we have one more question there.
Yeah. Marina Savelyeva, Novartis. I'm one of maybe few pharmacometricians here in the audience. Very good question, and Kostas, I think we discussed a lot about that, but we also had some discussions around this question with Flora a couple of months back at an EMA workshop. And, you know, for me, and she agreed, that's very important, is to really assess the uncertainty around your parameter estimates. Because, you know, as pharmacometrician, we start with very early data, like phase 1, even before that, and that certainly will be large. But as we learn and add more and more data, perform pooled analysis on the way, we hope to reduce this uncertainty. And what Flora said, that this comment was kind of unusual coming from a pharma, because really we do think, I strongly believe in it, we have to communicate this uncertainty very clearly.
Yeah.
And this is really something that, you know, no one solution, it depends. But if you say, this is what I know now, and this is uncertainty about how I estimate, for example, the drug effect, then I think it becomes very transparent.
Yeah, exactly. And I think, machine learning can have great advancements the last years in this post-selection controlling of the uncertainty with conformal prediction, with great frameworks around type 1 error. And someone can say it's exploratory, but still, they provide guarantees in type 1 error control. So if you put it in some sort of framework, I think you can even bridge these two nicely. Okay.
Thank you very much.
Thank you very much.
Thank you.
Yes, thank you.
Then we go to the last question. It's an from the field of oncology, but I think still kind of relevant to other fields as well. For example, in chronic diseases, there is also different, a treatment regimen has different stages or phases or whatever you want to call them. And how do we evaluate the efficacy of each of them?
Oh, sorry.
Okay, apparently I have the same problem with Mike as Mark did. So yes, thank you. Firstly, thank you very much for giving me this opportunity to present our problem and asking questions to all the regulators. So my topic is today is contribution of sequence. Sorry.
Yes, this works.
Oh, yes. So this topic comes from one of my study from oncology, non-small cell lung cancer. But I believe this could be also applied to other indications with multiple treatment phases. So many studies involving in oncology designed to involving the surgery inside the design, and there are different period of treatment period. Like the terms are very difficult to remember, such as like neoadjuvant, adjuvant, or perioperative. But I can explain what are these. So here is the diagram for the perioperative treatment settings. So patients coming to the trial and then randomized to either the experimental arm or control arm. Firstly, they will be treated by the treatment with certain cycle, and in this period, we call it neoadjuvant. Then after this neoadjuvant, patient will be going through the surgery. So basically after the surgery, post-surgery several weeks, then patients will continue the study with the same treatment with the adjuvant period. So this design involving the sequence of treatment actually having some concerns. Firstly, it is unable to isolate the contribution of the additional treatment effects in the different treatment period. And secondly, it could be causing the additional safety issue, and some patients maybe also get overdosed because they got too much treatment. So FDA actually held an ODAC meeting on 25th of July to discuss of this problem issue. So they clearly mentioned that they would recommend the multi-arm study for the different treatment sequence. They would prefer to have a factorial design with the four arms, including the perioperative arm, which is our design, our tested treatment, and then they want to also include the neoadjuvant only arm and the adjuvant only arm comparing to the control arm. So because this is a lot to test, and lead to a big scale study, so FDA have also said if it is not possible to have a 4-arm design, then a 3-arm design is also acceptable, including the perioperative arm, the neoadjuvant arm, and the control arm. So we have our questions. I hope everybody understands the scheme. And then, so the first question, I think a bit sharp, that FDA recommended a multi-arm study design. We would like to hear about the recommendations and the opinions from other regulatory agencies. And so I will just go through my questions. So assume that we decided to pursue a 3-arm design instead of 4-arm design to including everything. Then which type of treatment regimen would you prefer to be included in this study to compare with the perioperative arm and the control arm? And secondly, if so is this multi-arm study should be conducted in the phase 3 confirmatory setting, or is it also okay to conduct in the phase 2 proof of concept setting? So then this is that was the first big question for the new studies that we want to design. And for the second big question is for the already existing studies, because there are quite many existing studies with only 2 arms comparing the perioperative and the control. And so we would like to ask, is it possible to demonstrate the treatment effect using of different treatment arm to a different treatment phase using advanced statistical analysis methods? For example, we have some proposals like landmark analysis, and we analyzing from a certain time point like a surgery, or indirect comparison with using the external cohort or historical data. Or we can also maybe use some exploratory analysis on the responders or subgroups. So the last, we want to hear about the guidelines.
So thank you. There's a lot of questions. But let's see. Whoever can start, wherever you feel like starting with these many questions.
Maybe I can try to make a start, but I'm not sure whether I can answer all the questions, honestly. I maybe have to say that oftentimes we only see the 2-arm trial, unfortunately. That makes it quite hard to assess the contribution, as you've mentioned and FDA has mentioned, of the separate phases. So I think any approach to try to tackle this is helpful. I don't really see how we could do it in a 2-arm trial to establish relevant information on, especially on the contribution of your adjuvant phase. So for the neoadjuvant phase, I do understand that during surgery you can assess how successful this treatment phase was, but then it's difficult to understand what the added benefit is, because we have a differently pre-treated population. And so we don't know what is like just carrying over and from the success of the surgery and what is then from the adjuvant phase. What you could do, rather than to have a 3 or 4-arm trial, you could re-randomize. That would be a simple and standard solution, and then you would address a slightly different question. It would not be the whole phase, the whole trajectory over the whole treatment, but it would be the question when I completed my surgery and successfully, then what added benefit do I get when I'm treated in addition or afterwards again? And for me, that would be the most natural question, because also in a patient trajectory, I don't decide whether I will get treated perioperatively, but it will be decided I get an neoadjuvant treatment, then I get my surgery. Then the question is, do we need to continue treatment? So for me, the re-randomization would be a more natural choice than the studies the FDA has proposed.
Okay.
In the studies, I have to say that with the 4-arm trial, you can nicely pool your treatment arms, at least in the first phase. So you can pool the arms which were randomized to placebo, you can pool the arms which were randomized to treatment. And so you have a quite high power during the first phase, and you lose a bit of that power in the second phase. And because you don't re-randomize, the question then might get different than to what you do when you do re-randomization. But other than that, I think having more information on the understanding of what contributions to the different treatment phases have to your success would be very beneficial for patients and healthcare professionals. And I think hence it's a very good discussion we have here today.
Okay, thank you. But thank you very much for this re-randomization advice, because we also got the same advice from other experts that we probably can do this re-randomization after surgery. But I personally have a question on the re-randomization, because then that means after surgery you do re-randomization, then the patients will be diluted the effect, because they change. Some patients would go from the adjuvant to the control, and then the power will be reduced. That was my thinking.
It depends on your assumptions, doesn't it? I mean, that's also the question of whether I can pool or infer anything from the second phase. It's true. If I believe that you need a whole treatment cycle, then you would see a stronger effect in patients who were treated in the first phase and the second phase with your product, rather than patients who were just treated with placebo and then randomized to the active arm. But that you can also you can infer it from your data. So maybe you cannot test it, or maybe you don't want to test it, but you still can infer it, and you can nicely distinguish the contributions of these different treatment schedules.
I have a question. Is there any difference between re-randomization and just doing a 4-arm trial? Does it really matter when I randomize?
I believe, yes, you have got another experiment at the stage after surgery. So you might die before that, or you might have a bad outcome. So you cannot be re-randomized. So you might have additional inclusion-exclusion criteria. So you change the patient population and also over time. So it answers a slightly different question, but it answers a different question, I believe.
Plus, the outcome of the surgery could be an intercurrent event that is actionable for the next treatment decision.
Andreas?
Yeah, I think this is really very complex, and I basically agree to all what has been said. But I think with this phase 2 study, I cannot imagine that the endpoints that we typically use in phase 2 will allow us a meaningful conclusion, because I think many of the concerns that come from this 2-arm study, where we cannot disentangle the two phases, come from safety. That we have long-term toxicities, and we are wondering whether it makes adjuvant treatment makes sense. And I think this is really difficult to weigh with phase 2 endpoints. And also in general, I think we should not forget that the motivation between all this is safety, or many parts.
Okay.
We didn't comment on the last question yet. There is a guideline on the contribution of single agents, and the default recommendation that is in there is pretty consistent with what I understand FDA has recommended. So I rather understand your question as how can you leverage some of the information from the phase 3 trial? And that, of course, depends. The joke never gets old, kind of. And it's really not an easy question. Also, in Scientific Advice Working Party, this is something that comes up repeatedly. And I don't think that we can easily give a very clear answer whether this can be solved in phase 2 or by making one of the arms smaller or something like that. That really depends, and it's not easy.
Agree, yes. And so for the existing 2-arm trials, is it because there could be some same study but with different period, like actually in BMS we are having the kind of sister studies, like the first study is in the neoadjuvant period only, and then patients go through surgery and they stop. And then the second study is for the perioperative treatment comparing to control. So that was my question, that is that acceptable to do the inter study comparison to address the adjuvant treatment effect?
I think it also depends. It is a really complex question, but it also, I think, depends on the targeted indication, right? The claim that you then want to make out of all of this, because I think when it then comes finally to the approval, it really, the indication also really matters. What do you think that you have the evidence for? So maybe that is also something, you know, for the team to consider there. I don't know if Eva has any thoughts around the indication, basically. Will the indication be that then your drug will be approved for this and that, like as one regimen, or will there be one line which says for this neoadjuvant and for that adjuvant kind of separately, right? That also makes a difference. What is the target?
Yeah, I can just echo what you said. That definitely makes a difference.
So does everybody who starts the neoadjuvant treatment is from that point onwards supposed to get the whole thing, right? Or whether there is, like Benjamin said, an interim decision after the operation to decide whether to continue, because then maybe different evidence is needed.
Yeah.
Okay.
Maybe, I mean, that's a discussion of the treatment attribute of your estimand. So maybe that already introduces the first session, I think, later today.
Yes.
So thanks all the panelists, everyone who presented. We now break for 90 minutes. So I ask everyone to be back at 2:45. Lunch is outside, and also